In this last session, the third and the final session, we will look into the third major experiment, as a result of which the scientific fraternity was able to, with a definition, with a great resolute, conclude that the genetic material is DNA. And this experiment was conducted by two scientists by the name Martha Chase and Alfred Hershey. And here we have these two individuals. And their experiment is also known by another name. It's called the Waring Blender experiment. Now, Waring is the brand of is a brand. It's the name of a company which produced blenders, which still does produce blenders, the mixie jars that you've seen. So this is that blender, all right, which was used in their experiment. Now, what was the aim of their experiment? What did these two individuals, what were they looking for? They were trying to figure out which of these two molecules is the definite contender, or is responsible for carrying the genetic information from one generation to the next. So let us have a look at their experiment. What did they do? Now, Martha and Chase, both of them, they realized that a certain difference in the chemical property of proteins and DNA. Now, proteins have sulfur in them, and DNA has phosphorus in them. There is no phosphorus in proteins, and there is no sulfur in DNA molecules. And this is the distinction that they made. So, to start their experiment, what they did was, first of all, they generated proteins which were labeled with a radioisotope of sulfur. So first of all, they obtained a radioisotope. So I hope you know what an isotope is. Basically, forms of an element or an atom with the same atomic number but different atomic masses, right? And this particular isotope of sulfur has an atomic mass of 35. This particular isotope of phosphorus has an atomic mass of 32. And the speciality of both these isotopes is that both of them are radioactive, and that's why they are called radioisotopes. When I say radioactive, I mean to say that sulfur 35 and phosphorus 32, they emit radiations, they emit high energy radiations, and these radiations can be detected by using certain instruments, for example a Geiger-Müller counter or an ion chamber. So there are different devices that can detect their presence. So the first step, what these guys did, they understood the basic difference in the chemistry of proteins and DNA. And what they did is, they first of all obtained these two isotopes. They obtained the sulfur isotope and they obtained the phosphorus isotope. Now, in their experiment, what were the material that they were using? So they used a bacteria called E. coli, and they used the radioisotopes. Let me just write for the short form. And the two radioisotopes that they used were the sulfur 35 and phosphorus 32. And the third important component that they used was a bacteriophage. And what type of bacteriophage they used? They used a coliphage. Now let me just come back to this. A bacteriophage is a virus that infects a bacteria. Now, even the virus have specificity. They are a mammalian virus. They are a virus of the plants, animal kingdoms. There are fungal viruses. They are a virus that only infect bacteria. And the virus that infect bacteria and cause harm to them are called bacteriophages. And there are again variety of various types of bacteriophages. So there are these bacteria called the E. coli bacteria, or coliforms. We call them the coliform bacteria. So one of the coliform bacteria is E. coli or Escherichia coli. And the virus that infects E. coli are called coliphages, right? So basically, this was the third content that they chose. Along with this, they chose a lot of other material. They used a lot of other material, chemicals, and certainly the blender, and we call it the Waring blender. Waring being the brand of the blender. Now, before we proceed any further into the experiment, we need to understand how the virus infects a particular bacterial cell. So what I'm going to do is, let me just draw a E. coli cell. So Escherichia coli is a bacillus. All right, so it's a bacillus. It's a rod-shaped bacteria, and this particular And here let's just draw this particular virus. So this is a coliphage, right? And what is this coliphage made up of, or this virus made up of? It is entirely made up of protein. The outer part is purely protein, and in the center we have the genetic material, and this genetic material is the DNA. So coliphages have two components, the protein coat and the genetic material, which is DNA in case of coliphages. And here we've got the bacteria, the E. coli. So let us first of all understand how this virus infects this particular bacteria. So the viral particles, which are, I've drawn both of them nearly the same size, but the virus is extremely small. It is like 1,000th and 1,000th part smaller, maybe even more smaller than a particular bacteria. So this particular bacteria, this particular virus, coliphage, it first of all goes, and what it does, it attaches to the surface of this bacteria. So this is what the attachment is like. So here we've got the viral particle which has attached itself to the surface of the bacteria, and here we've got the genetic material. And after a little while, once the connection is secure, once the phage attaches fairly strongly to the surface, to the wall of the bacteria, what it does, the second stage is injection, during which this genetic material is injected into the E. coli bacteria. And what do we also have? The E. coli has its own genetic material, right? And here is the DNA of the E. coli. And after a certain period of time, what happens is the DNA from the inside of this particular coliphage is sent inside the cytoplasm of this bacteria, and eventually this particular DNA fragment of this coliphage then eventually integrates itself. It basically puts, let's use a yellow marker for this coliphage, and it integrates itself into the genome of the E. coli, right? So let's rub this green color, and the yellow color is representing the DNA of the virus of the coliphage. So now this is basically how a particular infection of the cell takes place. So the genetic material is sent inside, the protein coat remains outside, and after this a lot of other things happen. The cell is hijacked by the DNA of the virus, and the cell starts to make protein coats, etc., and a lot of other things, and then eventually the cell breaks up and the viral particles are released. But we don't need to understand that or know that at the moment in greater details. What is important for us to learn here is that the protein coat stays outside on the surface, while the genetic material is injected inside, where it gets integrated into the genome of the bacteria. This is what we have to understand. Now, taking all of this information into account, Hershey and Chase, both of them, they designed this amazing experiment, and here is this experiment. But before we proceed into this, we will see what they did. So they designed two different types of virus. First of all, let us not get into the image right away, rather let us just have a look here. So what we are going to do is we'll use this particular part. So using this idea that proteins are rich in sulfur and the DNA is rich in phosphorus, or uniquely DNA contains phosphorus uniquely and proteins contain sulfur. Proteins have no phosphorus, DNA has no sulfur. So they made use of this difference, and what they did was they obtained isotopes, heavy isotopes of both of them. Now let me rub. So they obtained from a certain lab, from specialized laboratories, they obtained the two isotopes of sulfur, radioisotopes. So they obtained S35 and they obtained P32. Both of them are radioisotopes. They obtained them probably in vials or in certain containers. And then what they did was they first of all made a culture media to grow E. coli. Now remember, this is very interesting. This is how the actual experiment went. So they used a petri plate. They used many petri plates, and they prepared a culture media in which they had to grow E. coli. And in that culture media, in that particular nutrient, in one of the nutrients, they added the sulfur. The source of sulfur was this heavy isotope of sulfur. They did not add the normal sulfur there, the normal isotope of sulfur, because that was not radioactive. They added to this culture media the radioactive isotope of sulfur in the nutrient media, and then they added some E. coli to it, just a couple of little bit of E. coli, so that they can consume this culture media and grow large number of colonies, right? Similarly, they took another petri plate, and in that particular petri plate they also had more. They prepared the nutrient media, but in that nutrient media everything was normal. All the entire culture media had the same nutrients, but the only difference was that the phosphorus that they added in this culture media was radio labeled, was this heavy isotope. They did not use the normal isotope. And then they took some E. coli bacteria, and then they inoculated this culture. They added it to this culture. So what happened after a period of time? So let us now just see the bird's eye view here. So this is the bird's eye view of the first plate. This is the second plate. So after inoculation, they started getting these colonies of E. coli. There were a few seed bacteria which were used like a seed, and then they grew into colonies. And now what was special about these colonies, we'll have a look at in just a little while. And in the other plate also the same thing happened, right? So we have like two plates in which we grew E. coli. How do these two plates differ from each other? Both of them have the same nutrient media, same food for both the E. coli, but in the first plate the source of sulfur is radioactive, and in the second plate the source of phosphorus is radioactive. So now the E. coli here has radioactive sulfur, and here the bodies of the E. coli has radioactive phosphorus. Now these particular plates were infected by coliphages. Both of these plates were infected by coliphages. These particular virus, I hope I'm still audible to you. Now let us have a look here. Yeah, now this is a coliphage here, right? And both of these plates in which we had grown the E. coli, they were infected by coliphages. Both of them were basically compounded with the presence of coliphages. Now the coliphages went and infected all of these E. coli cells, right? Hijacked these cells. So let us say this is one of the cell here. Is an E. coli cell. Let us say which has taken up, which was grown in this particular plate, the second plate. The second plate was rich in radioactive P32. So this particular, where will you find this P32 now? You will, because this is unique to DNA. Let me just rub this a little bit. Let's draw the bacteria absolutely again. Let's use a black marker to draw the bacteria. So let us say this is the bacteria, right? And the DNA will take up the radioactive phosphorus, right? So let us draw the DNA with the blue color, right? Now anything that contains phosphate, anything that has phosphorus, whether it is in the cytoplasm or in the DNA, all the phosphorus will be labeled by P32, this particular isotope. So once this particular virus, the coliphage, attaches itself on top of it and hijacks the cell, it will start to build its own copies. It will start to make virus inside. So let us say this is the inside of the cell. Let us draw the inside of the cell. Let us say this is the inside of the cell, a little bit of inside of the cell. And inside of the cell here, let us say this is the DNA of the same bacteria. This is the DNA of the virus. Now this virus genome has hijacked the entire cell. It will start to make shells. It will start to make the protein shell. So let us make the protein shell. So the protein shell is all black. But the protein shell, will it contain any phosphorus? It will not, because proteins do not contain any phosphorus. And what is the where is the virus getting the raw material from? It is getting it from the cytoplasm. And the cytoplasm also has radio labeled P32. So now once the virus starts to make its own DNA, when it makes its own DNA inside and packs it inside the cell, this DNA will be radioactive. It will be radioactive. Why will it be radioactive? Because DNA contains phosphorus, and the cytoplasm of this particular E. coli cell, because it is grown in this particular media which is rich in heavier isotope of phosphorus. So the DNA of the virus now that will be prepared by using phosphorus, which is radioactive, and the DNA will become radioactive, whereas the shell is made up of protein, which is non-radioactive. So we are able to obtain radio-labeled coliphages. Why did they do this big massive step in the first place? The first step was performed, this first part of the experiment was performed to obtain radio-labeled coliphages. So I hope this much is clear to you, and let us move now. Let me just clear up this particular area and let's talk further. Now the first step was conducted to obtain radio-labeled coliphages. Now the radio-labeled coliphages were grown in two separate plates. Plate 1 had sulfur 35 and plate 2 had phosphorus 32. Now as a result, the radio-labeled coliphages, the ones that were grown in plate number 1, had a radioactive protein coat. I want you to think about it. In plate 1, we have sulfur 35. Sulfur can only be a part of proteins. So now this media here contains sulfur 35 protein. The E. coli bacteria take up, all the protein components become radioactive. Now when you grow the virus inside, the virus will make their protein shells. And protein contains sulfur. So where is the sulfur coming from? From these coliphages. And from these coliform bacteria. Where did these coliform bacteria, E. coli, got their radioactive sulfur from the media? So the protein coat that they have is radioactive, but the DNA that they contain is non-radioactive. Why is it non-radioactive? Because DNA does not contain sulfur 35, right? It has no sulfur in it. There is no sulfur present in the DNA. So from plate 1, Hershey and Chase obtained radioactive coliphages. What kind of coliphages? Coliphages that had their protein coat radioactive. Their DNA was normal. The DNA was normal. Likewise, if we look at the second plate here, this is the second plate here. The second plate led to the production of radioactive coliphages, which have a radioactive DNA. Why does this plate have radioactive DNA? Because this plate is rich in phosphorus 32, which is a radioactive phosphorus isotope, right? And this is phosphorus is only present in DNA. And these particular coliphages took this particular P32 from the E. coli bacteria that were grown. Now I hope you also know that all the viruses are obligate parasites. They cannot be cultured on culture medium. They need living cells to grow, and that is why E. coli cells have been grown. So far in this first phase of experiment, the objective of the first phase of experiment was to obtain radioactive coliforms, two different forms of radioactive coliforms. And now I'm going to rub this off and make a little more space for myself. So now the experiment, the second phase, as I'm calling it, I don't know whether this was the second or the third phase, but by this time now we have got radio-labeled coliforms. And we've got two radio-labeled coliforms, and now let us use the color. We have got here the first. So they isolated these coliforms now. From plate 1, they separated all the coliforms. From plate 2 also they separated all the coliforms. They purified all of those coliphages. I'm sorry. They purified the coliphages from plate 1 and they purified coliphages from plate 2. And they separated them and put them into two conical flasks like here. Now this flask here, let us look at the first flask here. This is the batch 1, and this particular flask contains virus. Which virus? Coliphage. Now what is special about this coliphage? This coliphage is labeled with sulfur, which means that the protein coat, now whatever I'm going to draw in red, is radioactive. So the coliphages present inside here, the protein coat is radioactive and the DNA is not radioactive. These are the coliphages present in here. And in the second flask, what do they have? They have coliphages with a normal protein coat, non-radioactive protein coat, but the DNA is radioactive. See in red, it's radioactive. Why is the DNA radioactive? Because of phosphorus 32. I hope you are still connected. So now they get these two radio-labeled coliphages, and now the experiment actually begins. And what do they do in this particular experiment? Is that they take a petri plate. Now here is the petri plate. Now why are Hershey and Chase doing this experiment? Let us recall once again. Hershey and Chase are conducting this experiment to find out which of the two is the genetic material. Is DNA the genetic material or is protein the genetic material? Let me get myself a new slide, a clean slide, and let's start. So what do we have? Now let me just draw a bit. So I've got a radio labeled coliphage with a radioactive protein coat and a normal DNA, non-radioactive DNA, and we have another set of coliphage which has a normal protein coat but a radioactive DNA. Now also let us write why is this protein coat heavy, because this contains sulfur 35 and this contains phosphorus 32. This contains phosphorus 32. Yeah. Now what do they do is, here they take a normal culture plate. So these are normal culture plates, and inside both of these they take two separate. So these are two different lines of experiments going side by side. These are two parallel experiments running side by side. And in this first particular plate, in the nutrient media, this is a nutrient media to grow E. coli colonies. So let us say these are E. coli colonies. These are the colonies, bacterial colonies of E. coli. And again we have the same kind of a plate right here, right? So we have plate right here. Now what we do is we introduce this particular radio labeled virus inside. In the first plate, we also introduce in the parallel experiment, we also infect the second plate with the radio labeled virus. Now what is going to happen after so this is basically the infection stage. Now what happens inside? Let me just draw a virus. So we have a let's go back to the diagram where we have it, and let me just magnify it. So let us say this is the part where the bacteria is being infected. So this is the radio labeled protein coat. So here we've got the coliphage in which the coat is radio labeled. So this is the radio labeled coat, and the DNA is non-radioactive. This is here inside this particular jar. Now these particular bacteria are then these particular E. coli bacteria are infected with this particular virus, and what happens is that these virus, they go and attach to the surface. So now have a closer look. We are just demonstrating one of the bacteria. So what happens that these particular viral particles, they go and attach to the surface of this E. coli bacterium, a normal E. coli bacterium, and they insert their non-radioactive material inside. So the DNA is inserted inside. So once this DNA is inserted inside, let us have a look at the other image. So let us use this blue marker. So let us say here this material is also sent inside, this material is also sent inside. So after a little while we have material only on the inside, right? And we don't have any more DNA of the bacteriophage present in the bacteriophage. What we have, we only have shells on the outside, right? It's something like this. Let me just run. So after infection, in the first case, we have got this bacteria, and we only have the shells. We only have the shells of these particular virus, empty shells attached onto the outside. So these are the shells which are attached onto the outside. And where is the DNA? The DNA is lying on the inside. And so this process, the injection of the DNA, it basically occurs within the first 20 minutes of infection. Within the first 20 minutes of infection, this is the condition. And what these individuals, what did Martha and Chase do after this? After a certain period of time, as far as I can remember, it was approximately 20 minutes. After 20 minutes of incubation, 20 minutes after they added the virus to the bacterial culture, this was the condition. All of the bacteria, most of the bacteria had the protein coat, empty shells on the outside, and their DNA on the inside. Now, if you bear in mind that the coat, radioactive the protein coat, the shell is radioactive, right? And now they added this entire solution, this entire bacterial culture. See, so they had this bacterial culture here and they added the virals to it. After 20 minutes of infection, they took this entire solution and they added the solution into a jar, into a blender. And this is the blender. And then they turned the blender, they started the blender. And because of that high force on the inside, the viral particles, the protein coats were sheared off. They were thrown apart. They broke off from the cell. So now after really mixing it up, everything really well, because of this extreme shearing forces, the bacteria was separated from the shells. Now inside the bacteria we had the DNA, which was non-radioactive, and then we had the protein shells. Now after a certain period of time, after a certain period of blending, which probably was a couple of minutes, which I'm not too sure about how much that was, after a certain period of time, they stopped the blender and they transferred the entire solution into a test tube. And once they transferred it into a test tube, they centrifuged the tube. Now, what is centrifugation? I hope you know what a centrifuge is. Basically, centrifugation is the process in which we put tubes in a particular machine, and then that particular machine, it rotates, it spins at very high speeds, and the contents in the tube are, according to their weight, they sediment down, right? So the heavier particles will sediment down. So the next step was that this particular blend, this blend was centrifuged, and as a result of that, because see, the bacteria are larger and much more massive than these viral empty protein radioactive shells, all the bacteria, because of that centrifugal force, it settled down and it formed a pellet. Okay, so it formed a pellet. It was a thick solid sediment at the bottom, so that's a pellet. So what is inside the pellet? Inside the pellet we have all the bacteria, and inside the bacteria we have the DNA of the bacteria plus the DNA of the virus, right? So all the DNA of the bacteria and DNA of the virus is here, and the supernatant liquid, all of this liquid, it contained the empty protein shells. How do we know that? Because we know this because we are aware of the differential sedimentation of these particles. The lighter particles stay up and the heavier particles, they sediment down. Now what they did is that they exposed this tube to a Geiger-Müller counter or a radioactivity detecting machine, and that particular device, it detected the radioactivity in the sample above. The radioactivity was not found in the pellet. The radioactivity was found in the suspension. And what did Hershey and Chase infer? They inferred that the heavier content, which means the cell which contains the DNA, is at the bottom, and the protein, which is a part of the shell, is outside. And hence, see, the genetic material has to be inside the cell, and there is no radioactivity coming from the cell. This means that the protein never entered the cell, and the protein is not the genetic material. And what is the genetic, what could be the genetic material? Something else, which we will see in the other line of experiment. So at least in this line of experiment, what did they conclude? That the radioactive proteins never entered the cell. They never entered the pellet. They remained outside. So from this line of experiment, they could only conclude that at least proteins never went into the cell, and if they never went inside the cell, if they were never a part of the pellet, and inside the pellet were the bacterial cells, if they never were a part of the cell, they've never entered the cell, then they probably are not the genetic material, right? Now let us look at the second line of the experiment. The second line of experiment is here, and the second line of experiment was going parallel. It's the same kind of an experiment. In the second line of experiment, they had isolated these coliphages which have a radioactive DNA, and let us draw it here. So now what kind of a coliphage is there? They have this particular coliphage, non-radioactive protein shell. The shell has no radioactivity because these coliphages were grown in media that contained P32, radioactive phosphorus, and phosphorus is a part of DNA. And so their DNA is radioactive, right? So this is the second line of experiment. What they did in this second line of experiment is the same as the first. They again took a Petri plate inside of which we had normal E. coli colonies, and those E. coli colonies, they introduced this particular suspension of radioactive coliphages with radioactive DNA to them. And once they were introduced, again after 20 minutes of incubation, like in the first line of experiment, this is what happened. Let us look at the second step. This is what happened. The same thing. The viral particles attached themselves to the surface of the bacteria, and they started introducing their DNA inside the bacterial cell. And now, because since we've used the red color to represent the radioactive DNA, this is what was happening. So the DNA was getting inside the cell, and the experiment was run exactly in the same way as the first line of experiment, the one that we just saw. And after 20 minutes, when entire radioactive DNA was inside the cell, when infection had completely taken place and the shells outside were empty, the entire solution, this particular entire content, was transferred to this warring blender, and the blender was started. And because of the shearing forces, what happened again? The same thing. The shells went off. They got detached from the surface of the E. coli bacteria, and then we got this slurry. We got a slurry, and after 20 minutes, after a certain period of blending, we stopped the thing. We transferred the entire solution into a tube, and the tube was like really cloudy, and we exposed that tube to a centrifuge machine. What the centrifuge machine does, once the centrifuge machine is started, it spins at a very high speed and pushes heavy particles down. So now which component is heavy? The cells are really large, much more dense than the viral particles. So the cells settle down and form a pellet. Right? Here is the pellet. And then we have the supernatant liquid. And then they expose this tube to a radioactivity detecting system, maybe a Geiger-Müller counter or an ion detector or something of that sort, some kind of a machine. And what they detected was, they detected no radioactivity in the supernatant liquid. What did this mean? This meant that the shells have no radioactivity. But where did they detect the radioactivity? They detected the radioactivity in the pellet. And what is in the pellet? The pellet has the cells. And why have the cells become radioactive? They become radioactive because the radioactive DNA has got inside these cells. So now this experiment confirms that the genetic material, now which is the genetic material? DNA in the virus was the genetic material, that the genetic material is the DNA and not the proteins. If protein was to be the genetic material, it should have been inside the cell, right? Because that is what the virus do. Viral particles, what do? They move their genetic material inside, the host cell. I hope that this particular experiment is clear to you, the warring blender experiment or the Hershey and Chase experiment. And so the conclusion of this experiment again turns out that proteins are not the genetic material, but DNA is the genetic material. I hope this is clear to you. Thank you.